Our final speaker this morning is Dr. Iain Croall. Dr. Croall received the 2021 Beyond Celiac Established Investigator Grant along with his colleague, Dr. Nigel Hoggard. Dr. Croall is a postdoctoral research fellow at the University of Sheffield, whose research examines how gluten-related conditions affect the brain. Dr. Croall and Dr. Hoggard's 2021 grant builds on their previous work that showed evidence that people with celiac disease have a greater risk of damage to brain white matter, as well as worsened cognitive and mental health. And at our November mini conference, Dr. Croall showed us some very cool slides with spinning brains, and he's assured me that he's going to show them to us today as well. Welcome, Dr. Croall.
Good morning, everyone. I'd like to thank you all for being here, and obviously also thank you very much to Beyond Celiac for inviting me to be here and to present this work. So I am going to be talking today in quite a general sense about, this interaction between celiac disease and the brain. And then I'll be moving on to describing a couple of experiments which kind of demonstrate some of these points, and then finishing up talking briefly about this exciting new collaboration which we have with Beyond Celiac. I am going to be talking primarily about celiac disease today specifically, but I often find it very useful to begin these talks by kind of extending the scope of that idea a bit further to also include this idea of neurological problems which are still happening due to gluten sensitivity, but which don't necessarily need to occur alongside celiac disease at the same time. So around the early noughties, a couple of these phenotypes were defined, and we have a dedicated clinic at the university and at the hospital that we collaborate with for people from all over the UK who have these conditions. These are relatively rare presentations, but they're still very important and useful to kind of get an orientation point on. So on the left here is what we would call gluten ataxia. There's a number of different forms of ataxia. Ataxias are disorders of the part of the brain known as the cerebellum, which for various causes undergoes a large amount of atrophy, and this causes lots of problems, and the cerebellum is kind of essential in processing of movement and coordination. So in this particular scan of someone who has ataxia due to underlying gluten sensitivity, we can see that their cerebellum is very much not as big as it should be. There's also another phenotype called gluten encephalopathy, which doesn't necessarily involve the cerebellum so much. It involves the top part of the brain, the cerebrum, and doing another kind of MRI scanning, we can see a lot of what we would call white matter lesions, these blobs, which in this particular scan are actually very severe and very expansive, that essentially should not be there. The symptoms that we expect to see as a consequence of these reflect quite cleanly the parts of the brain which have been affected. So with the cerebellum being involved, as expected, we see patients who have difficulty walking, movement and coordination. Some of these patients end up wheelchair bound. The more that the cerebral white matter is involved, the more we see issues of cognition, foggy brain, bad headaches, these sorts of things. So the way that I like to think about this is that there is a kind of a single broad phenomenon, which is gluten sensitivity. This can affect the gut, it can affect the brain, and it can also, of course, affect the skin as well. But rather than these being completely clean individual diagnoses that are separate from one another, what we really have is a spectrum of kind of interplay between them. They can affect one system to a clear and obvious degree, and when it does, we can give a name to that. So obviously, if the gut is affected very badly with clear enteropathy, we call that celiac disease. If the skin is affected, we call that dermatitis herpetiformis. And if the brain is affected to a substantial enough degree, we can call that gluten ataxia or encephalopathy. There's been a bit of talk today about serology, and of course, we're developing these different serological tests, which they're certainly good for associations and maybe point a little bit to potential causes. When we're talking about tissue transglutaminase in celiac disease, these are antibodies against TG2 we're looking for, which have almost kind of 100% sensitivity and specificity for the condition. TG3 has been fairly well implicated in dermatitis herpetiformis, and at Sheffield we've been developing, in collaboration with the University of Cardiff, I believe, and investigating over the past few years antibodies to TG6. These do not seem to have the same really powerful sensitivity and specificity. They don't seem to be completely diagnostic of people who go on to have gluten-related neurological problems, but there does seem to be a very clear kind of overrepresentation in that group. So this is something which we're looking into more and trying to kind of see the scope of that and how far this takes us. So for the next bit of the talk, I'm going to give you a really quick crash course in some different imaging techniques, which obviously I find very interesting. What can imaging help with here? Well, a really clear thing to say is that it can obviously help with patient care and diagnosis and sort of tracking progress over time. Again, this is a fairly kind of extreme example, but it demonstrates the point quite well. This was a patient who was diagnosed with gluten encephalopathy. And you can already see in the baseline scan that there are some white blobs and splotches that stand out that should not be there. This patient did not want to go on a gluten-free diet. They had a follow-up appointment 2 years later, and there has been a very extreme development of those white matter lesions over time. I believe after this, they did then go on the diet, and progress of this seemed to slow down and stabilize after that point. They had a lot of this is diagnosis by serological testing, diagnosis of exclusion from other things as well. And we do often, I'm not a clinical doctor myself, but my colleagues who do treat these patients, they very commonly see these stabilizations and improvements after the diet is engaged with. So this is an example of what I would call a structural scan. A structural scan is probably what everyone thinks of when you talk about imaging. It looks like a picture of the brain. Both of these on the screen at the moment are structural scans. On the left, we have what you'd call a T1 MRI scan, which is kind of your gold standard, just looking at the actual structure of something. It's just a decent anatomical picture. The one on the right, where we get these white matter lesions, that's now a T2 MRI scan, and physics properties and magic of magnets and radio waves involved in that mean that white matter lesions become very bright on these, so you can kind of use a T2 scan to explore if these types of pathology are present. We can then get into types of scans which are a bit less intuitive, but perhaps actually even more interesting. So we can do something with an MRI scanner called magnetic resonance spectroscopy, where instead of getting a picture, we get these things that almost look like seismographs. And there are peaks that happen on these seismographs at different locations, and based on the height of those peaks, that tells us the concentration of a specific metabolite in the part of the brain that the scan has been directed to. So I've highlighted 3 on this seismograph here, or I should say spectra, really. The one on the right, called N-acetyl aspartate, is a particularly interesting one. This is something which kind of reacts to a number of different problems that can affect the brain. You'll usually see its levels go down after the event of some sort of insult or injury. It's thought to reflect either neuronal density or neuronal metabolic activity. More is better. And we know from experience that if you measure N-acetyl aspartate in the vermis of the cerebellum, this is also part of the diagnostic process we use for finding people with gluten-related neurological problems. It's very dependably found to be very lowered in that area. And also, this is from a publication from 2017, you also then see a recovery of N-acetyl aspartate in those patients who go on the diet and follow the diet. And then it's actually quite a nice clear pattern. Those who refuse to go on the diet, the level continues to decrease over the time, and it appears relatively kind of stable in people who show evidence of having followed a partial diet. We can, here's the spinning brain, the colourful spaghetti. You can also do what you call advanced imaging. This is from a kind of scan called diffusion tensor imaging. The raw data of this doesn't look anything like a brain. It's something that you feed into specialist software that fits mathematical models and all sorts of things on top of that. What DTI is really good at doing is imaging the brain's white matter, and I keep banging on about white matter here, but it's interesting in this context. The white matter is essentially the wires that run through the center of your brain. They really do look like wires. They're these kind of nerve fiber tracts. They connect all the grey matter together. So you can do a DTI scan and process it in such a way where we then actually can kind of form what these wires look like from a particular person. So this is from a patient with celiac disease, and to my knowledge, it's the first time this technique had ever been used in the study of that patient was when I created this. If you want to do experiments on it numerically, though, it doesn't look quite as exciting. These kind of blobs on the right-hand side of the screen are from that. You can kind of distill the scan down to different metrics and biomarkers, and then compare how those numbers shift between two groups or change with different outcomes. And I will shortly get to an experiment where we did this to explain that a bit more. So lastly, before I talk about these experiments, I just want to address what is always a bit of like an elephant in the room with these things, which is general acceptance and awareness of how the brain can be involved in gluten sensitivity. As a point of interest for myself, I tried to find the oldest reference in medical literature that appeared to link the two together. So I went on PubMed, and this article from 1966 by Cook and Smith I found, where within that they themselves refer even further back. So as far back as 1908, there is a publication, case report type thing, 2 patients with what was then known as sprue, with peripheral neuritis also in the legs. And then there was also a report from 1925 of a patient with sprue and ataxia and anesthesia of both legs, which matches up extremely nicely with our descriptions and understanding of gluten ataxia today. Moving forwards, there's certainly been progress from that. People have done research beyond what I'm going to be presenting today. There is a reasonable body of evidence now, that these things exist, and there is some progress that's been made with that. In the UK, we have this healthcare body called NICE, who basically write the gold standard reference points for patient care and treatment for any and all conditions, essentially. So if something you've done makes it into the NICE guideline, that's a real sort of well done sticker, that you can get. And NICE guidelines now do directly reference that celiac disease involves a number of non-GI symptoms that can involve neuropathy and ataxia, and also now outright recommend that if you have a patient with otherwise unexplained neuropathy or ataxia, it's the recommendation to do serological testing for celiac disease. So this is very good, but it's also only one half of that coin that I've been trying to talk about. This still frames everything around celiac disease specifically and the serology and GI presentation of that you'd also expect. It's not yet acknowledging this can still be due to gluten, even if the person is also not diagnosable with celiac disease necessarily. This kind of ongoing debate was summarized, I think, quite nicely by this systematic review, which was published in 2019. They essentially looked throughout the literature for any reports that reported prevalence of ataxia and neuropathy in cohorts of celiac disease patients. For neuropathy, the prevalence estimate started at 0% and went up to 39%. And for ataxia, it again started at 0% and went up to 6%. So these 0% papers, within their own little world that they've created in that experiment, they are essentially saying these do not exist. These are not kind of risk factors associated with it. The conclusion of that report, I thought, summed things up quite nicely, where they wrote, although that these neurological manifestations have been described much more than 30 years, as I've just been presenting to you in the literature, there are still diagnostic delays that can result in permanent neurological disability. These are attributed to controversies which arise from variation in reported prevalence and poor understanding of the use of appropriate serological testing. There's a couple points here which I think they really nail. First of all is the need to get these diagnoses correct, because the brain does not recover from injury in the same way that the gut can do. We can kind of learn our ways around some injury that's happened and show some improvement, but generally speaking, things that physically happen to the brain stay the same way afterwards. So it's important that people who would benefit from treatment, the gluten-free diet and so on, are identified at an earliest point as possible and given that advice. And secondly, this latter sentence kind of gets to what I think the heart of the issue is here, which is not quite understanding the right way to try and identify these problems. And this is particularly pertinent in the literature because, this was a systematic review of outright ataxia or neuropathy. These are quite blunt problems. If you have full-blown diagnosable neurological ataxia, you are fairly progressed with the condition already to have reached that point. But there is a large spectrum of neurological health that exists before that, and a lot of the framing of this often seems to go, do you have ataxia? No, well, we're not interested. But there's a whole range of things that happened before that point which is still relevant to the brain and is absolutely relevant to the patient's experience of the world as well. It's a very common anecdote of someone who kind of feels that something's not quite right within themselves, but the medical tests don't quite show that you're bad enough yet to be referred appropriately and so on. So the first experiment I want to discuss is one which was published a couple of years ago, where we attempted to validate this issue in patients with celiac disease. So we got access to a study we have in the UK called the UK Biobank. For anyone who's not familiar with this was a giant undertaking by the Wellcome Trust. to recruit 500,000 people who essentially just had to be an adult in the UK of any and all healthcare background. And then over time, there's all sorts of healthcare data that's been collected about these people, and they're now, I think now they're actually quite close to the end of this undertaking to perform various types of MRI scans on 100,000 of these people. So needless to say, within that number, inevitably there will be patients with celiac disease, and we have brain scans and cognitive data for these people as well. And importantly, to kind of, preemptively shield against criticism, which can sometimes be leveled at these experiments, which do show that, these findings are there. This is independently collected data, not from our own centre. These are going to be patients who are fully representative, who are population of celiac disease in the most pure definition of population science. And, the data collection has not been done with celiac disease in mind at the time either. So the question essentially becomes, can we see these sorts of findings in other people's data? We identified volunteers with celiac disease who were otherwise healthy. There was a very long list of exclusion criteria and other conditions. They were not to be taking any medication which could affect the functioning of the brain at the time of data collection, and they also had to have brain imaging available as well. And there were 104 such cases in this cohort at the time when we conducted the experiment. We then matched them in a 1:2 ratio to healthy controls on a range of different criteria. Not listed here, but we did also check that smoking frequency and alcohol intake were also the same between two groups, and they were not significantly different either. So we're trying to control for vascular risk factors here as well, which can also affect the brain in similar ways to what we were then going on to be studying. And we then just wanted to perform a series of case control analyses. We just wanted to see if there's any evidence that the celiac disease group were a bit on the back foot in terms of, the brain scans or the cognitive performance. So we looked at key scores from 5 different cognitive tests. We looked at responses which were given to 6 different questions which explored aspects of mental health. And we also did some brain imaging analyses, the center point of which was using this kind of advanced imaging. This is a very good thing to use, not just because it's colorful and looks pretty. It's very popular within all sorts of neuroimaging research. So, it's everywhere in dementia, in Parkinson's, in stroke. It's a very kind of well-adopted scanning technique, and it is very popular because the measurements we get from this are very sensitive at picking up on kind of relatively early physical changes to the white matter of the brain, which are maybe not yet detectable on other forms of imaging. And it has very good reproducibility as well compared to some other kinds of imaging also. So in terms of the results, we did indeed find that the patients with celiac disease in this cohort had slower reaction time significantly compared to the match controls, and they were significantly more likely to give responses to these multiple choice questions that explored quality of life and mental health. And those were true of the questions looking at anxiety, depression, thoughts of self-harm, and perhaps not quite so surprisingly, how happy they were with the state of their own health. In terms of the imaging analysis, we did then find that one of our metrics from the diffusion tensor imaging was significantly different in a number of areas in the brain between the two groups. So what we're looking at here in the background is kind of like a template brain, and anything that's green is showing kind of the core of a white matter tract that is not significantly different in measurement between the two groups. The green areas are the same, they're not of interest. But anywhere that has a kind of like a red to yellow blob is a location where this measurement is significantly changed. And this is taken to show a kind of like a ground truth physical difference in the structure of the white matter between the celiac disease patients compared to the controls. And when you have this alongside a deficit in things like reaction time, it builds, I think, a very clear picture of, a physical shift, which has led to a tangible cognitive outcome as well. And we were very happy that this got published in a very, reputable, quite high-impact journal, and it also received an editorial at the point of publication. So in the spirit of this, trying to kind of, validate the issue and increase acceptance and awareness, we saw all of that as being a very kind of positive uptake from that study. So with it established, hopefully, that this is an issue that we should be paying attention to, I'm going to talk briefly now about what actually are the symptoms we might see in patients with celiac disease, how frequently do they occur, and is there anything we can look at which might predict risk of these happening in some patients more than others. So this is now another experiment which was published a couple of years ago, but which was using data from around 2015, I think. We recruited 100 patients with celiac disease from our gastroenterology department. None of them had any history of neurological disorders, and they were all recruited at the point of their initial diagnosis as well. So the point of all this is that there's a real effort made to get, a typical, if you will, celiac disease group at the earliest point of their diagnosis, at a very early point in their disease course. All subjects, we just looked to characterize the cohort. They all underwent a neurological examination. They self-reported problems they felt that they had. We did blood sera testing for a variety of things, but including this transglutaminase 6, which we're wondering might be a bit of a predictor for gluten ataxia. And we also did some brain imaging as well. In terms of the symptoms, the red blobs are what was found on clinical examination, and the blue blobs are what was self-reported. There's a variety of different things that seem to fall down the lines mostly of balance, headache, and sensory symptoms. So 10% had signs of peripheral neuropathy, 29% were noted to have some level of kind of difficulty walking, and 11% had nystagmus, which is like an eye twitch sort of problem you can induce on examination. And then in terms of self-reporting, they kind of matched up quite nicely. A quarter of the group reported some difficulty with balance. Headaches were very frequent. And lastly, there were some sensory symptoms. And I think a nice summary for this is that even after you exclude the headaches, because headaches are very non-specific, you could still say that exactly half of the group had at least one sign or symptom of possible neurological dysfunction here. In terms of the brain imaging study, I then wanted essentially to see if those patients who also did have TG6 antibodies on top of the diagnosis seemed to be at any additional risk of kind of neurological complications. There wasn't much that came out from this in terms of looking at how symptoms fell down these lines, but the brain imaging was very interesting. So 40% of these subjects did have TG6 antibodies, so it's almost half of the group also this was true for them. And sure enough, those that did had lower brain volume in a couple of regions in the brain. So first of all, in the cerebellum, and then also that as a general effect, that is across all of the cerebellar grey matter. That's where that number was kind of taken from. And then we also did kind of like a first pass investigation looking at the total volume of subcortical grey matter, which are grey matter regions in the top half of the brain. And we did find an effect there, and when you looked at that more deeply, looking at which subcortical region is it driving that, it was the thalamus. What's of interest here is that in the previous experiment I was discussing, where we got these white matter tract differences, they were very much focused on the tract that connects the cerebellum to the thalamus from the biobank data. And then in this study, we're finding apparently the two regions which might be at risk of atrophy are the cerebellum and the thalamus as well. So we're starting to build a little bit of like a synergistic picture between these different experiments now. Lastly, experiment 2.5, we did a follow-up on this data much more recently. So how do these symptoms change over time? Is there any evidence that the gluten-free diet can help progression, which I'm sure is something we're all very interested to know about at these sorts of talks. So talking about COVID interfering with things, it was meant to be 50 of these patients who reattended, but there was a spanner in the works in various different ways. We wound up having 30 of these patients who reattended a mean of 7 years later after that initial assessment. And they essentially underwent all the same investigations again. The scans, importantly, were performed on the same scanner using the same sequences, which is always a consideration that needs to be made for these things, because small differences in hardware or software can produce artificial sort of differences in results. And with it already being established that the cerebellar grey matter and the thalamus are regions of interest, the volume of these areas were kind of reassessed at both time points. The differences between them were taken, and then ultimately that number was kind of split by the actual length of time that had passed between the two scans for each patient. So the number at the end of it is a yearly percentage brain volume loss for the thalamus and for the cerebellum. That's what we're going to be looking at. And just at the bottom is for my own kind of nerdy interest for considerations that need to be made when picking your software packages. Everything in imaging is really dependent on having good templates for a starter. So you want to use this package on the right called Suit, which is dedicated to doing cerebellar imaging. So exactly half of the cohort had achieved negativity of all gluten-related antibodies. I think in an ideal world, you would want to look at how differences progress over time by each individual antibody. But with the relatively small sample size we have, we were kind of forced here to go, well, let's just create two groups, half of which still have at least one antibody present and half of which who have achieved total negativity. Progression of symptoms over time is a real mixed bag. So needless to say, the baseline percentages are the baseline of this 30, not the baseline of the 100, from the previous experiment. A couple of things appear to get better. Headaches are less frequent now. Nystagmus is apparently a bit better. There was one fewer person that happened in. But just as readily, some things appear to have worsened over time as well, and a couple of things appear to have been stable. We can try and interrogate this a little bit further for patterns. This isn't something I want to spend too much time on because this is not a statistical finding, but you can kind of descriptively note, for example, that all the new patients who were reporting balance issues who did not have it at a baseline were all also patients who were still positive for at least one gluten-related antibody. So there's possible trends there, but definitely things we'd want to kind of interrogate more thoroughly with bigger data sets. We were able to show, however, I was very excited by this, in the brain imaging, there was nothing in the thalamus, but the rate of atrophy in the cerebellum was indeed greater, statistically speaking, year to year, in the half of the patients who were still positive for an antibody compared to the half which were not. So in the group of patients who had achieved negativity of antibodies, they were losing 0.32% of cerebellar volume in the grey matter a year. And this was over double in the other group, who were losing a mean of 0.73% each year of the cerebellum. This is very exciting for me because there are a handful of studies out there, kind of like demonstrating these kind of statistical differences. But this is the first time, to my knowledge, that brain imaging has been used to show a tangible benefit, apparently, of strict following the gluten-free diet, at least to the point of achieving immunological negativity, which I know is the objective of doing that this does appear to confer a benefit. Of course, what's missing from this is a control group. We're not able to say that it's completely arrested progression, just that it's better in one group than the other. It would be great to also have a control group to add to this slide as well. So that's a bit for future research. So just to bring it all together, brain and cognitive changes are detectable in patients with celiac disease, even when we use third-party data and population data. This really helps validate that this is a problem and is something which needs to be kind of fully adopted and accepted, I think. The sorts of symptoms which might be expected, neurologically speaking, include headache, foggy brain, some coordination, some sensory problems. These mirror quite nicely as a kind of a microcosm of the sorts of things which we see at a much more severe level in these rarer cases of people who have very severe, kind of strictly neurological presentations of these, which kind of raises questions, I think, of, all this sort of shared disease mechanism. Perhaps are celiac disease patients, quote unquote, lucky in receiving a relatively early diagnosis and then going on a diet which helps things not get worse? All good questions. And finally, there is evidence that a gluten-free diet does help this progression over time, but more research is needed, which lastly brings me to the collaboration which Beyond Celiac has very generously started with us. I mentioned this clinic which we have where we see patients who do have these extreme neurological presentations. Over the years, we've had over 1,000 such patients. In the course of their regular clinical care, we'll have multiple scans from multiple different time points, serological tests galore, notes on electrophysiology recordings. We're also collecting new data as well. There's a postal questionnaire on diet adherence and mood, which has now been getting posted out, and that's going to form the basis for an abstract to the Celiac Disease Symposium at the end of this month. Ultimately, we want to combine all of this and do some really high-powered, in-depth analyses by disease subgroups, by serological results. How does all of this really progress over time? What factors ameliorate things in the middle of this? Ultimately, is the gluten-free diet enough, and how does it affect the patient's quality of life at the end of it? So, thank you very much all for listening. Thank you also to my colleagues back home. That's Nigel in the middle, who unfortunately couldn't be here today, but also Marios and Dave, who kind of take the lead on the neurological and the gastro side of this research as well. And thank you all very much for listening as well.
Nice presentation. Is the implication that the anti-TG6 antibodies are crossing the blood-brain barrier and causing the white matter lesions? And if you believe that, have you made arrangements to get autopsy brains from these patients to actually look for deposition of antibody or infiltration of inflammatory cells into the white matter?
Yeah, absolutely. That's a question which, needless to say, we're very interested in at the moment. The short answer is we don't know exactly what the link is. We can see these associations between positivity being overrepresented in some groups and others. There are a number of different hypotheses we could go down in how all this transfers. So as an enzyme, TG6 is expressed throughout the brain and also in particularly high concentrations in regions like the cerebellum and also the thalamus as well. So theoretically, the idea of these antibodies crossing over and doing some kind of harmful cascade after that point, that is a reasonable theory. There's other things which I'm quite interested by. Some of the other gluten-related antibodies kind of on paper also show reactivity with different brain structures. So for example, gliadin antibodies have been shown to have reactivity with the blood vessels that supply the brain. And a lot of the white matter lesions that we see, my previous job was in vascular dementia, and it's quite striking to me that the presentation of people with celiac disease who develop these white matter lesions is a really close pattern to what you see in these kind of cerebrovascular conditions like vascular dementia. And there was a very large study done using Swedish epidemiological data, which did also show that although there is not an increased risk of patients with celiac disease to go on to get Alzheimer's or other forms of dementia, they do have an increased risk of developing vascular type dementia. So I think there's potentially a number of different routes at play here. There's also the potential of the body just being generally in a kind of pro-inflammatory state. That's also not good for the brain too. Some research has been done finding depositions of these in certain cells. It's not something like the cell biology side of things, to be completely hands up. It's not something that I'm completely comfortable sort of discussing at length, but these are absolutely things which we're also engaged in back in Sheffield as well and very interested by. The university is currently doing some research on brain scanning and long-term COVID symptoms. I'm not aware of any hypotheses yet which are linking that with celiac disease, but yeah, the brain fog is a commonly reported thing in a number of different conditions, and I think how that is also fascinating to me, how that works on a physiological level as well. Thanks. Yeah.
I had a follow-up question on brain fog specifically. So have you or anyone in your group been able to look at brains of individuals who report brain fog separate from COVID? And if so, are there any changes that you see there?
The closest that I can give that answer, we've done a fair share of studies, I mean, as Beyond Celiac has recently, showing that brain fog is a really commonly reported symptom. There was a pilot study that I was involved in a couple of years ago, where we recruited patients with non-celiac gluten sensitivity. And we did a questionnaire on about 125 of these, but we asked 5 of them to come in for brain imaging studies as well. We're trying to do a very novel sort of experimental setup where they all volunteered to gluten themselves and have a before scan and an after scan, with the after scan being timed to try and coincide with when they said they would expect symptoms to begin. This is an N of 5, so I'm not saying this is anything to sort of like really hang your hat on. One of the patients who did then go on to experience brain fog, one of the scans we did appeared to show a very large spike in cerebral blood flow to the brain at the same time as this. It's something which I've been interested in trying to follow up with since, but currently that's not unfortunately something which has got further traction. But it's always stuck in my mind after we did that as well. Thank you.